So this session is protecting the Great Lakes region and coastal ecosystems from air pollution. And the session co-chairs are me. I'm Alex Free from Minnesota Sea Grant, and we're based at the University of Minnesota Duluth, and Trent Wickman, who you just heard from. And Trent will be our first speaker. So Trent is at the USDA Forest Service, and his talk will be partnerships and monitoring the Northwoods Lake Monitoring Project.
All right, I'm excited to talk to you about a lake project that we did in 2022 that I think is something that I'll always be proud of, even when I'm sitting in my retiree chair somewhere thinking about this project. I think it's something that's going to be a baseline that people are going to use for years to come. So I have a number of co-authors and a number of partners that pretty much could be co-authors. We'll talk about that, but this was a very partnership-oriented project to look at lakes in the Northwoods, which is probably the cleanest, well, is the cleanest lakes in the area. So for state agencies, a lot of times they focus on the problem areas and where the pollution is. And so it's interesting that we got all the states to jump in on this and really focus on areas and lakes where a lot of people would think, oh, that's good. We don't need to worry about those ones. So just a little bit of orientation to the EPA National Lake Assessment, because this is a add-on to the National Lake Assessment. So EPA has National Aquatic Resource Surveys, or NARS surveys, where every 5 years they rotate around, and they have one for lakes, they have one for small streams, they have one for wetlands, large lakes, and I think coast, is that the other one? And you rotate every 5 years around. So the first lake survey was 2007. I've been involved every year. They've had these surveys because it's basically EPA does a random draw of lakes. And so wherever the lake they pull out of the hat is, you got to go try to get there and get a sample. A lot of those end up on the national forest. So I was helping coordinate with the states and EPA to get those samples previously in previous surveys. And as somebody who's interested in water quality, even though my primary position is air quality, I mean, people in this room understand why the two are related. And I'm very interested in understanding how air quality relates to or influences water quality. And so I've always been trying to pull together water quality data across the 3 states that I work in Minnesota, Wisconsin, and Michigan. And anybody that's tried to do that, know it's a really fun thing to try to do that, because you have different surveys that have happened at different times, using different methods to answer different questions, and trying to put those all together and make sense of it is really a career-long project, because I'm still working on it. So that's sort of the background for why this opportunity is a great opportunity, because it's one where EPA sets the standard and we all collect the same way across not only this region, but across the country. And it's done statistically in a tiered way that you can tier up. So you can compare your results to the nation or other parts of the country. So it's very valuable. Specifically in this area, what we were concerned about is, we want to really characterize the water quality in this region, not only as a region as a whole, but also within the region, is how do we understand differences, even though we think that everything is similar in some ways. There are differences across Minnesota, Wisconsin, and Michigan. Some of the stressors we're particularly concerned about are climate change, increases in dissolved organic carbon, which is we call browning. There's a lot of papers I've talked about in the Northern Hemisphere, increase of the DOC, and DOC is natural in our lakes, but what we're seeing is it's increasing over time. We have an issue with harmful algal blooms, and especially in wilderness areas where you would have no other watershed type kind of source for it. So trying to understand why in our wilderness areas we're seeing algal blooms. And then you have typical things like residential development, mercury we've been looking at for years. So want to look at, so what are the ecoregions is what the terminology that EPA uses. The smallest ecoregion in the standard survey is the upper Midwest ecoregion. And one of the things, again, people that live here forget about it, but we have 50% of the natural lakes in the US, non-reservoir lakes, in this area. So that's pretty obviously a nationally significant resource. So it's interesting to bring that up to leadership and remind them what we have here. But when you look at this map, and if you're familiar with the Midwest and the upper Midwest ecoregion is not really a great area to use for somebody that's interested in the Northwoods, because not only does it include the upper forested area, but it also includes agricultural central hardwoods area. And so when you mix that together, you get results that aren't really useful. So the states and I got together and said, hey, how about we carve out just the Northwoods and try to do our own survey and tag in? So if you want to tag in and do a smaller area, obviously statistically, you need to collect more samples. And so what we wanted to do then is take the map on the right, that's just the Northwoods, and then also split that into what we'll call sections and try to understand the differences within. And so that's the approach that we took. So within that whole area, there's, what is it, 30,000, over 30,000 lakes, greater than 1 hectare. So, and then we took and looked at, we weren't going to be able to do, roughly with the stats would say you got to do 30 lakes in each area you want to define. And so if you took that with all those sections, it would be just a crazy amount of lakes. But what we did is figured out the 7 most lake-rich sections and just focused on those. And that was 83% of the lakes in the whole Northwood. So that worked out pretty good. So 30 lakes times 7, and then with the lakes that the EPA would already was going to do with the national survey, we end up with 250 lakes. So we have 250 lakes in 2022 in the summer in 3 months that we're going to sample. What are we sampling? Typical stuff that you do with a limnological survey. Go to the deep spot in the lake. So we don't do the complete, people that are familiar with NLA, we didn't do the complete survey. They have a shoreline survey, a sediment survey, lots of other things. We just focused on going to the deep spot, the index site, and do the chemistry and typical field measurements like Secchi depth and profiles with a sonde. We also had, when people heard that we were doing this sampling effort, everybody wanted to give us a bottle to go collect something for them too. And so we had to kind of be careful about that. We did 2 add-ons. One was isotopes, and then one was colored dissolved organic matter at the University of Minnesota. So as I go through the results, I just want to remind you of the ecoregions the EPA uses, because I'm going to refer to some of these other ecoregions as I compare the results. The ones that come up are Northern Appalachians, which will be NAP, Southern Appalachians, SAP, and Coastal Plains, along with Western Mountains or some of the other ones, the ones that we're going to talk about. So this is kind of how they're going to look. If you look, this first graph on the left is Secchi depth. So Secchi depth is a little disc you put it on in the water, and when it disappears, that's the depth. So the bigger the Secchi depth, the clearer the water. And if you look on this graph, what we start out with here on the far left is US. So this is this total 2022 survey average of Secchi depth. This is meters. So you can see that compared to that, the upper Midwest is clear, which should not be a surprise compared to the whole US. We have the 3 states here also that we put in, Michigan, Minnesota, and Wisconsin, just for reference. But Minnesota, those 3 states do additional sampling so that they can have data for their entire state. And if you remember from that map, some of these states include the agricultural areas, and Minnesota actually includes the tall grass prairie. So those are really including some productive areas that are non-typical. So that's why they kind of bump around. NLF is our data. So you can see that we're pretty darn high on here. We definitely are clearer than the upper Midwest. And the only people that are clearer than us are the Western Mountains. So that's this one here. So they're the clearest of the whole country, but we're second clearest. And then if you look at chlorophyll, there should be sort of an opposite relationship here, because chlorophyll is an indicator of algae. And so here's the US again, upper Midwest is less. The 3 states kind of bounce around in there. You can see northern lakes and forests were less than upper Midwest and much less than the country. And the only place that beats us is the Western Mountains. Let's see here. All right. Nitrogen is something that I'm really interested in as an air quality specialist now. It seems like we've won the battle with sulfur. I'm interested in nitrogen. We're not very far from that nitrogen bomb down in southern Minnesota and Iowa that's changed our deposition from oxidized nitrogen to reduced nitrogen now that we're looking at. So this is total nitrogen. So not an actual super cool comparison to that, but it's definitely, like I said, interested in nitrogen overall. And you can see that, like some of the other pollutants we're going to talk about, we have less nitrogen here than in the US and the whole upper Midwest. But actually, there's some other parts of the country that have less than we do even. The coastal plains, southern Appalachians, northern Appalachians, and of course the Western Mountains. But getting back to the nitrogen and nitrate, for example, nitrate is very rarely measured in the lakes around here. The only time we measure nitrate above detection level tends to be in the spring or the fall, because these are nutrient poor systems. So any nitrate or ammonia that falls in the system is sucked up immediately. So just a note here that out of our sample in northern lakes and forests, only 8 lakes out of 240 were measured above detection limits. That just goes to show the point that right now, the landscape's not seeping out any nitrate. And if it ever did, that would be a sign that something has changed potentially. Acidity, fun one this group will get a kick out of. Again, countrywide here at 8, upper Midwest a little bit higher. Again, these states include a lot of ag regions, so that gets them higher. Our northern lakes and forests, we are about 7.4. Northern Appalachians win. They're the lowest. I don't know if that's a win or not, but again, not surprising. And then these other two are higher than us, coastal plains, Western Mountains. EPA in their National Lake Survey has criteria for what they call natural acidification and unnatural acidification, and it has to do with DOC. I mentioned DOC is hugely important here. If you have high levels of DOC, that's naturally acidic. So I was looking through the data to see what lakes we had that might be non-naturally acidic. So low DOC, but also low ANC. And we only found arguably 3 or 4 lakes out of our 240 that met this criteria. So you go back to some of the programs that EPA had for lake monitoring that included obviously the Appalachians, but also included up here in the upper Midwest. And these lakes that did pop out are in areas where traditionally had been assumed to be the more sensitive areas potentially for acid deposition. So again, they're still out there, but they're just not very many. Sulfate's really important. Randy talked about that for anybody that was here yesterday. We've got a history of declining sulfate in lakes that go right along with deposition. But another thing that we have here in Minnesota is this wild rice that is very important to the tribes. It's really in their story of how they came to the areas. They moved and moved from the east until they found the place where the food grew on the water. That's a reference to wild rice. So it's sacred to the tribes. And so sulfate is a byproduct of mining and also wastewater treatment plants. Minnesota has a standard for 10 milligrams per liter for sulfate that was fought about, and it's been fought for many years, but currently is the standard. And you can see that the 95th percentile value for sulfate across all of the Northwoods lakes is 1.6. So to have anything anywhere close to 10 is considerably an outlier. And it's interesting when you look and find the few that are outside 10 or higher, they're almost always related to a mining operation, either current or historical mining operation. And I just wanted to put up, this is some different data. This isn't from the NLA. This is some data that forests have collected over the years. This goes all the way back to the 80s. Some of these lakes, we've taken advantage of previous lakes that were EPA LTM lakes. But this, on the top of this graph, you can see these are 3 different NADP sites with sulfate. And then these are the lakes below. And you can just see they mirror each other. If you look at the percentage decrease of sulfate in the lake versus sulfate in the air, that's just basically, they just follow each other right along. This is an amazing success story. We talk about things that we need to be able to talk about to the public. This is one. I mean, we're not going to, I don't want to talk about chemical and chemistry, but to talk that we can make changes to the atmosphere that make real changes directly to the lakes. It's right here. It's really amazing. And this is the same graph for some lakes over in Upper Peninsula, Michigan. We also have one for Wisconsin too. Chloride, just to mention, anytime we have chloride that shows up at any significant level, it's usually related to proximity to a road, and it's road salts. And states are more and more interested in this increasing amount of chloride in lakes. And what does it really mean? And lastly, this is my Jason Lynch comment for him. Our 50th percentile value for DOC in our lakes is 9.3 milligrams per liter. 50th percentile. We have a lot of DOC in our lakes, and it's super important when we're trying to understand the chemistry of our lakes. I think too often we take things from like the Appalachians and other models that were developed and just try to slap them up here. We have a completely different situation here. We need to be able to deal with the DOC in a real way and not just kind of an approximation. So what's the future? This is just real quick. We just got this data back. It was some data wrangling because it came from multiple labs, but we got it all put together. And so what do we want to do in the future? Want to understand how our lakes are unique. We want to understand the spatial patterns like I talked about. And what are some good predictors for those patterns? Two different categories: local characteristics, different things like geology, soils, land use, all the typical things you can see there. That would be local and regional factors. I'm interested in atmospheric deposition, especially related to nitrogen. Like I say, I think we're going to declare victory on sulfur and just leave that one now. And also trying to understand landscape features and climate, which are related to those ECS sections that I mentioned before. And I just want to end with a few fun pictures. The training that we did for this before in that 3-month sampling was in Wisconsin. So of course we went to a cheese and brat shop. And then this is some training on the right here where we were out with tribes, the people that were going to help with the project. This is at Trout Lake Station in Wisconsin in a different training spot. Super fun for me as a limnological nerd to go to Trout Lake. And just want to touch on the partners. Again, this is really the main take-home point is we could never have done this. These are people that did this with no funding. I basically called people and said, hey, we have this idea. Do you think it's cool? And if you thought it was cool, do you want to help? We don't have any funding for your time, but we'll pay for the analysis of the samples. And all these people jumped on. So we have the main leads here with the state of Michigan, Minnesota, and Wisconsin. And then we have field crew leads that went out and did the sampling. A number of tribal organizations are in here along with the National Park Service. This is the Minnesota group here on this slide. The Wisconsin group is in the left table here. We again have some colleges, tribes, national forests. And then on the right-hand side is the Michigan group, again, national forests, some county soil water conservation district folks, and again, National Park Service folks. So the biggest thing I want for you all is to say, hey, that's really cool. I want to do some analysis on that data because this data is your data. And we're probably within a week or so from posting it online. We'll have it all cleaned up. And we really want you all to use it and tell us what it means. Turn this data into knowledge. So contact me and I can put you in touch with the data. And yeah, I think that's what I got.
We have 1 minute for a quick question. Does this one work? It does. Okay.
Trent, thank you. You mentioned harmful algal blooms, but you didn't choose the microcystin analysis. Was there a reason for that?
I'm trying to think of how difficult that would be to sample. I don't know the method of that to sample off the top of my head. We were trying to figure out what we could do with crews that were moderately knowledgeable, but not super knowledgeable. We had to train all these crews. We tried to get people that were hydrologists or fisheries biologists by training. So, but we didn't always get that. So a lot of times we were dealing with people that just had an interest. So we were trying to keep the sampling as low tech as possible. And I think that was a piece of it.
So our next talk is a recorded talk. This one will come from Daniel Runo from St. Lawrence University, and it's trends in nitrate, ammonium, and total nitrogen deposition in the Lake Erie Basin, a 20-year analysis. And
it is an honor for me to be invited to present analysis of atmospheric nitrogen deposition in the Lake Erie Basin, a 40-year analysis at this national meeting. My name is Daniel Ruano, and I am a geology major at St. Lawrence University. This summer, I did a research undergraduate experience at UConn with my mentor, Dr. Marina Astita. The main question I focus on my research is, what are the changes in nitrogen deposition in the Lake Erie Basin for the last 40 years? My main data source was the National Atmospheric Deposition Program, and I used the National Trends Network, providing long-term record of precipitation chemistry across the United States. These two figures show Lake Erie. On the left is a satellite image showing an algal bloom in the southern and western portions. And on the right is an elevation and depth map of Lake Erie, and the western portion is the shallowest, and the eastern portion is the deepest with its dark blue. This figure shows the difference between deposition, precipitation, and concentration. I focused on nitrogen deposition, of the amount of nitrogen over the area around Lake Erie, so kilograms per hectare year. Now moving on to the methods, I selected 5 monitoring stations around southern and western periphery of Lake Erie, and the main criteria was high quality data from the last 40 years from 1980 present. The stations are Chautauqua in New York, Kane Experimental Forest in Pennsylvania, Worcester in Ohio, Roche Lake in Indiana, and Kellogg Biological Station in Michigan. This map shows the 5 selected sites. For the annual criteria, they were characterized as meeting the criteria or not meeting the criteria, valid or invalid, and the trend line was over a 3-year moving average, only when the minimum data are met for a 3-year period. Moving on to the results, the same format will be followed for each of the 5 sites. The top graph shows the change in nitrates, the middle shows the change in ammonium, and the bottom shows a change in the total nitrogen. The units are kilograms per hectare year for all of the graphs. For PA29, the nitrates showed consistent decrease, a high of 26.8 and a low of 6.5. The ammonium showed no significant change, more cyclic, and the total nitrogen decreased with an apparent trend of a high of 9.2 and a low of 3.3. For MI26, for nitrates, there was again a consistent decrease with a high of 21 and a low of 6. For ammonium, there was no significant change and it appeared cyclic, and for total nitrogen, it fluctuated without a significant trend, with a high of 9.2 and a low of 4.4. Now moving on to Ohio, there's very good data for this one. The nitrates show a consistent decrease with a high of 19.3 and a low of 6.4. The ammonium showed an increasing cyclic trend, and the total nitrogen fluctuated without a clear trend, high of 7.8 and a low of 4.4. Now for ion 20, the nitrate showed cyclic trends decreasing. The high was 19.3 and the low was 6.9. For ammonium, there was no significant change and it appeared cyclic. And for the total nitrogen, it fluctuated without a clear trend with a high of 9 and a low of 4.7. Lastly, NY10 showed the lowest quality data. For nitrates, the cyclic trends appear to be decreasing with a high of 26.3 and a low of 9.8. For ammonium, there was no significant change and it was generally cyclic. And for the total nitrogen, it fluctuated without a clear trend with a high of 10.1 and a low of 5.1. Overall, I found that there was a consistent decrease in nitrate deposition across all of the 5 sites, which are all in the periphery of Lake Erie. The New York site was the closest one to Lake Erie. The average reduction in nitrates deposition was 32%. And the largest reduction was registered in PA 29, and the lowest reduction was registered at the MI 26 station. As for the ammonium, it cycled at all the sites with no clear trends based on what I observed in my research. And the total nitrogen deposition decreased only at PA 29 to 36% with a negative 5.9 kilogram per hectare. And generally, the total nitrogen fluctuated in the other sites. There are many questions that came up during my time at UConn. For example, the change in nitrates that were decreasing can be due to the Clean Water Act, the Great Lakes Water Quality Agreement, and perhaps other regulations that have helped curtail pollution in the region. Also, the nitrogen deposition generally correlated with the precipitation. So if there's an increase in precipitation, it might change the overall data based on the precipitation more than nitrogen. And the regional depth differences with the lake is also significant because southern Lake Erie is shallower than the northeastern part and that has had more issues with eutrophication. But with nitrogen deposition, there didn't seem to be a significant difference, but I imagine for concentration, there would be to measure the lake water itself. For future research, there are many different areas that can be investigated. For example, federal and state level agencies that have an impact on policy and the best practices and nutrient management of public lands and protecting natural resources. Also, the aspects of the nitrogen cycle not being impacted now because of the variability in ammonium. And as far as the issues in the lake go, investigating why and how dead zones happen in Lake Erie will be important as climate change progresses and has a continued impact on Lake Erie. And lastly, this research really shows the importance of NADP to continue to conduct research and monitor all these sites so that we have good data to send to policymakers, which can make valuable decisions to preserve clean air, clean water, and continue to take care of our environment. There are so many people who, without them, it would not have been possible to conduct this research. I'd especially like to thank Dr. Marina Estita for being my mentor in the project and being an active researcher in Lake Erie, and also for Dr. Alexandra Hain. Lexi was a great help being the PI of the grant. And also for the overall research undergraduate internship was very valuable at UConn this summer. I'd also like to thank the National Atmospheric Deposition Program for hosting this amazing conference. And it is great to have everyone come together to discuss these important topics. And also for St. Lawrence for helping me present my poster at St. Lawrence as well. And if you have any questions, feel free to ask. My email is listed there, and thank you.
Now, so our next talk is going to come from Marta Vanier from Indiana University, titled The Ins and Outs of Per and Polyfluoroalkyl Substances in the Great Lakes, the Role of Atmospheric Deposition. So let me pull up her slides.
Good morning, everyone. What an interesting morning to be here and talking about PFAS. So first of all, thanks. This is my first NADP meeting, and I'm a little bit of an outsider because I'm not involved in the NADP, but I'm going to tell you a little bit about another monitoring network. The results I'm going to be talking about today are presented in this paper. If you're interested in a lot more details than I can give you today, we have the longest supporting information ever for this paper, probably of all of our papers. There's a lot of information that I'm not going to be able to give today. So I want to take this opportunity to give an introduction on the Integrated Atmospheric Deposition Network, in short, IADN. which has been measuring chemicals in the Great Lakes since 1990. So not as long as NADP, but fairly long too. IADN was born as a binational monitoring network between the US and Canada. And nowadays, it's not binational anymore, really, but we try to keep at least one collocated sample here at Point Petrie, which is our QA/QC monitoring site. We have locations at 2 urban sites, one in Chicago, one in Cleveland. Sturgeon Point is our middle of the road, like rural site. And then we have currently 2 remote sites, one in Sleeping Bird Dunes and one at Eagle Harbor. Their site at Bourne Island has been closed, so we currently don't have any sampling sites on Lake Huron. IADN was created to support the Great Lakes Restoration Initiative, and in particular the Great Lakes Water Quality Agreement, with the idea of restoring and maintaining the quality of water and the Great Lakes ecosystem. The goals of IADN are really to look at spatial and temporal trends of both legacy and emerging contaminants. And we are trying to continue long trend monitoring of legacy chemicals while also keeping our eyes open for emerging chemicals. A little bit of information about what we do aside for PFAS. I know that I promised I was going to talk about PFAS, but the network started with legacy chemicals, in particular PCBs, legacy pesticides, HCHs, DDT, chlordane, endosulfans. And with time, more and more chemicals were added to the list as new needs were emerging. Flame retardants, like PBDEs and halogenated flame retardants, were one of the latest groups of chemicals that was being added to the latest, and the group of chemicals that is keeping us on our toes and super busy. Sampling methods are kept constant. We really can't change anything in the way we do. We collect samples, process samples, even instruments, because we don't want to break the historical series, something that I'm sure you're very familiar with at the NADP. But we were able to start fresh with PFAS. And these are pictures of our site at Eagle Harbor, where you can see the rain samplers and the high walls. And this is Sleeping Bird Dunes. So particularly for PFAS and this presentation, I'm going to present results from precipitation samples. We collect 1 liter every 2 weeks from 5 sites. So we collect about 40 samples per year, and the results I'm going to present are from 2021 and 2022. We use the this sampler here, which I believe it's the same sampler that NADP uses, and it's rather just a glorified bucket, really. That's my definition of this sampler. This is what like a better picture of what the sites look like. We have here the PFAS sampler, all of the high-vol air samplers, and then the chemicals, which employ an XAD column that is then shipped back to the lab, and we discard the water. We don't do anything with the water for the legacy. For this study, we also collected open lake water. We worked both with the EPA and with Environment and Climate Change Canada to collect water samples on all of the lakes in spring of 2022 and 2023. This is a picture of how we collected the samples. This is actually the fancy sampler that was shipped to us from the Canadians. It's really like a painter pole with a container that is used to extend outside of the ship to immerse the water bottle. So very fancy, sophisticated technology to collect water samples. We also collected air sample using passive samplers. Passive samplers are really salad bowls. And when I say salad bowls, I mean literally salad bowls. We bought them from webrestaurants.com, drilled some holes, stuck a rod in the middle, and we put in between polyurethane foam discs impregnated with XAD resin. We deployed them at the sites for 30 days. And we collected these samples in 2021 and 2022. Because they stay out for longer time, sometimes you get surprises. Last month, we got a mouse into the sampler that was returned. And I want to mention, I'm going to go back to this, these passive samplers collect both particle and gas phase chemicals, which is very important. We analyze 7 classes of PFAS. We use SPE for cleanup, and we employ LC tandem MS for ionic PFAS and GCMS for neutral PFAS. So as Alicia mentioned before, these are the concentrations of PFAS in nanogram per liter across sites, ordered from the most urban to the most remote. And as you can see, there's no differences across sites, which is very interesting. And the first time I've seen this, I was like, that's weird. Can we go back and check the results? Because when you look at the legacy compounds that we have been measuring for a long time, there's a really strong correlation with the local human population. Basically, where people is, the chemicals are. And so you can see a definite trend. But this is not true for PFAS. So it seems like we're dealing with background levels that are similar across the Great Lakes Basin. If we look at the air, though, before we look at the air, these levels, I thought, since we were coming here, I wanted to compare our results specifically with those of the NADP. This is the paper from 2022, and I plotted here the most abundant compounds, PFOA, PFOS, PFHXA, and the penta, together with PFBA, which required a slightly different scale. And I want to point out that these are median concentrations at each of the IADN sites, and the NADP median concentration is the blue bar at the bottom. In general, I think the levels are comparable between the IADN and the NADP, which is always good and reassuring. PFBA is somewhat different. Our levels are higher. And it's not clear to me why there's a slight difference. I don't think it's a big difference. It could be due to different years in which the samples were collected. There's an increasing trend in the short chain PFCAs, so that might be part of the reason. But this speaks only also to the need for an intercalibration study about all of these precipitation networks. Now, if we look at concentration in water, here we have all of the lakes, and you can see that the levels are highest in Lake Ontario and lowest in Lake Superior, which may be due to Lake Ontario being downstream from Lake Superior, but it also relates to the degree of urbanization and human activities from Lake Superior to, which is the least populated, to Lake Ontario, which is the one of the most populated lakes. So we have a west to east sort of effect here. Now, it's interesting to compare the profiles between rain and open lake water. And as you can see, in general, the patterns are fairly comparable. And they are dominated, as was observed by Alicia, by the short chain PFCAs. The 4, 5, and 6 carbons are the ones generally dominating these profiles. We don't have air samples, precipitation samples on Lake Huron or Lake Ontario, so there's nothing really to compare to. Now, if we look at the total concentration in PFAS, we notice that the story changes a little. Concentrations are highest at the urban sites of Chicago and Cleveland, and they are lowest at the remote sites, which is what we generally expect for these persistent pollutants. The trends is still not significant, but when we look at the regression, as it would be for the legacy, but it's pointing that way. And it will be interesting to see what happens once we add more data to the series. Now, the profiles for the air samples were completely different from the water samples as expected, and they were dominated by the neutral PFAS, in particular 6,2-FTOH, which is more volatile. And the issue with 6,2-FTOH is that it can be degraded into the carboxylic acid that we know and we worry about. When we gathered all this data, the question is, so what? What does it mean? How important is atmospheric deposition in delivering PFAS in the Great Lakes? Is it an important mechanism or not? And so we tried to put it all together in what ended up being a huge effort, much bigger than we had anticipated going in. We thought we would do like a back of the envelope calculation. It turned out it was more complicated than this. So we put together in the output, we put sedimentation rate from the literature. We considered flows from water leaving the lakes using USGS data from tributaries and rivers. And from that, we subtracted the input of atmospheric deposition, which is wet, dry deposition, and air-water exchange. We included concentrations of PFAS from inflows, which is water entering the lake from tributaries, also using both flows and concentration from the USGS data, which is using passive samplers. And then we included also tributaries and wastewater treatment plants, again, from the literature. And I want to spend one more word on air, because it's really important. I also like air in particular. But I want to stress that the samplers that we use combine gas and particle together. And for this particular modeling effort, we had to account for the distribution between gas and particles. And so we use a partitioning coefficient from the literature that varies from 0 from the short chain PFAS, meaning that these compounds are mostly in the gas phase, to all the way to 1 for the very long chain PFAS that are almost exclusively on the particle. And so this is a very important aspect to keep in mind. So enough with the modeling details. What are the results? So there's a lot in this figure. I want to mention that when you see an arrow pointing upward, it means that the net flow is negative, meaning the lake is accumulating PFAS. When the arrow is pointing downward, it means that the flow is positive, and so the lake is discharging PFAS. When there's no arrow, it means that the flow is not significant. So let's look first at the colors, which indicates the specific pathway. So you can see that, especially for PFBA, which is the shortest of the 4 compounds for which we did this modeling effort, is mostly dominated by atmospheric deposition. And the role of atmospheric deposition decreases as we go to longer chain PFAS and as we move down water from Lake Superior. In general, these results suggest that the westernmost lakes, Superior, Michigan, and Huron, are still accumulating PFAS, while the eastern, I like to say north and south, but it's actually west and east lakes are still discharging PFAS. And there's indications that maybe Lake Ontario and Lake Erie may be at equilibrium for some of these compounds. Because I love legacy compounds, I thought it would be interesting to put PFAS into context. What does it mean in terms of comparison with the chemicals we've been measuring for a lot longer and that we probably know a lot more about? So here I put the concentrations in picogram per liter of PFAS compared with the legacy compounds, PCBs, pesticides, PAHs, and PBDEs. And you can see that PAHs, which are not particularly sexy anymore, as one of my colleagues once said, they're still dominating the concentrations in these samples. But PFAS are up there with legacy chemicals. Now, this is a teaser. When we put TFA in the picture, trifluoric acetic acid, then it becomes the dominating PFAS. We are working on TFA as a lot of the scientific community is too. The data are not ready yet. I could not show them quite yet, but TFA is the dominating PFAS out there, higher than PAHs and all of the other pollutants that we are measuring. So in conclusions, to wrap up, PFAS levels were homogeneous across the basin, so there's not a lot of impact from populated sites. PFAS level in water were highest in Lake Ontario and lowest in Lake Superior. The net flows were generally negative, so for Superior, Michigan, and Huron, meaning these two lakes are accumulating PFAS, and negative for Erie and Ontario, indicating that these are discharging PFAS through outflows or sedimentation. And we concluded that atmospheric deposition is an important source, especially for Lake Superior and especially for the short chain PFAS. One last slide to identify what are the gaps that we have encountered. These are research ideas for everybody out there of what we really need to have a complete picture. So we really need more data on discharge from tributaries and concentrations on PFAS. We really had one data point per tributary out there. The reviewers were not happy, I can tell you about that. We also need more data on ultra short chain PFCAs, including TFA. Going on the analytical side, it's not here, but we really need interlab studies to make sure that all of the data that we're putting out there are comparable with one another. We also need lake water samples from different seasons to account for stratification and temperature effect. And this is needed also for air and precipitation. We need to look at seasonal trends. We also need updates on sedimentation rates. I know that the sediment program is working and we're eagerly awaiting for those updated data. And then we really need to spend more time on understanding partitioning of PFAS in air. And not only because I love air, but also because the neutral PFAS are present at really abundant levels, and they can be degraded into PFCAs. And so at this point, it's not clear if the PFCAs that we measure out there in precipitation are really there from historical reasons, or they are there because they are degradation products of the very abundant neutral PFAS. And it's not until we understand better the partitioning and the abundances, we won't answer this question. With that, I'll thank the EPA for funding, and I should say a disclaimer, the EPA is not approved the TFA data, so that bar plot was not approved by the EPA. It's sort of a teaser, and thanks everyone for your attention.
Our next speaker is Samantha McClung. She's from the University of Minnesota Duluth, right up the hill. And she's going to talk about atmospheric deposition of PFAS, connecting wet deposited PFAS to observations in lake sediments. So.
All right. Hi, everyone. My name is Samantha McClung. I am a graduate student at UMD here in town, and I am going to share some of the work we're doing looking at PFAS and wet deposition and lake sediments. So a quick outline, I'm going to be going over our collaborators and then the basics of what PFAS are, how they're introduced to the atmosphere and removed, and then some of our preliminary data and wrapping up with some conclusions. So this project is pretty unique because we are working with a lot of different collaborators in order to incorporate large data sets that are existing as well as ongoing. And so I work with Dr. Alexander Free at the Minnesota Sea Grant, but we are also working with the NADP and Wisconsin State Laboratory of Hygiene, the EPA's Great Lakes Sediment Surveillance Program, as well as the University of Minnesota's Natural Resources Research Institute and Twin Cities Department of Soil, Water, and Climate. This project is USGS funded, and without the work of all these great people, our work would not be possible. So we've heard a couple talks already about what PFAS are, so I am going to kind of breeze through this. But in case anyone's just joining us, PFAS are per and polyfluoroalkyl substances. They're a very large group of man-made chemicals that are structurally diverse, and they are extremely stable in the environment and do not fully degrade, but have been known to negatively impact human health. And so our work is just looking at targeted PFAS, so those 33-ish different analytes, and it's difficult to talk about them all at once. So I've grouped them in these 5 categories that we have seen previously. And like Marta said, that some of these are precursors or they do partially degrade into our more terminal PFAS. So how are PFAS introduced to the atmosphere? This can be through a number of point and non-point sources. PFAS can be sorbed to particulate matter that gets airborne, or aerosols from the treatment of wastewater or leachate spraying, or even the use of aqueous film forming foams. Or they could just be desorbed from these particles or aerosols, or even our consumer products that have PFAS in them and enter in the gaseous phase. And so this image on the right-hand side is just an estimate of those potential PFAS contributors throughout the United States. But what this doesn't capture is that there are all these non-point sources, all of our consumer products that could be giving off PFAS at any given time that is not well understood. And so once these PFAS are in the atmosphere, they can be transported and deposited locally or long range and impact remote locations. And so PFAS can be removed from or via dry deposition or wet deposition. And so for the current literature that is available, wet deposition is shown to be more of a significant removal mechanism just based on the higher concentrations and fluxes of PFAS. But this is a very active area of research. And so dry deposition could be more important depending on the situation or location in question. And so our question is atmospheric wet deposition an important source of PFAS for lakes? And so the Great Lakes are right outside our window, and we just heard a great talk about how they are important. And so our original thought for this was looking at the water budget for the Great Lakes. You can see that precipitation accounts for 50% or more of the water coming into the Great Lakes for all of them except for Lake Ontario. So this just highlights that atmospheric deposition could be an important source of PFAS for these Great Lakes and something to consider when looking at pollution control. And so, like Marta just said, they see that it is an important source, and it really just highlighted for us that there's agreement on this issue and it's really worth investigating. So for our project specifically, we're looking at 3 different goals, and I am looking to determine chemical tracers of wet deposited PFAS, interpreting their chemical profiles to possible sources, and then assessing their importance to lake sediments. So let's jump right into our first goal here. So we are working with the NADP's National Trends Network sites, these 2 in Minnesota and 3 in Michigan. And these samples are collected on a weekly basis when possible and sent to the Wisconsin State Laboratory of Hygiene for analysis. So these 5 sites are either classified as rural or isolated based on the surrounding populations. And today I have about the first year of data to share. So this box plot just shows the sum PFAS flux of each sample collected at the sites. And I will note the very high outlier in Minnesota 23. I see he's there. We will talk about it more later, I promise. But right now, it's pretty variable across each site. You know, the interquartile ranges really overlap, but Michigan kind of might have higher amounts of PFAS, and this could just be due to population density in those areas. Michigan has more people, so urban areas generally having those higher amounts of ambient PFAS could be impacting this. Or this could be increased annual temperatures between the sites. We do see a slight correlation between the amount of total PFAS being rained out and the temperature, or even more likely, general increased amounts of precipitation seen between the sites. We see a much larger correlation between the two, and so this could just be an impact of the sites themselves. So of the PFAS that we're seeing, what are we seeing? The gaps between these bars are just due to times of either low or no precipitation, so the samples were either not able to be collected or composited. And so majority of what we're seeing are perfluoral carboxylic acids. We're hearing this over and over again this morning, and this is a trend that is seen worldwide. But there are these other times where other PFAS classes become extremely notable. Here in October, we're seeing an increased frequency of detection as well as abundance of these FASAs. And using the current literature that's available, these analytes are likely coming from either biosolids application or the spraying of leachates in agricultural practices. And so before I continue on with that, waste management facilities are not the problem with this. They are in a very unfortunate situation where they are receiving waste that contains PFAS, and their systems are not designed to either remove or destroy those PFAS. So I just need to say that I am not blaming them. So let's get into it. I'm going to focus on the Minnesota sites because I think their story is a little bit clearer. So this map is the agricultural land in Minnesota. All the different colors are different types of crops, where the white is either forested or urban areas. And biosolids application is generally done in the spring and fall for Minnesota. And you can see that Minnesota 23 is kind of smack dab in the middle of all of this ag land, where Minnesota 16 is a bit further away when you're thinking about the general direction of the wind here, which is west to east. And so Minnesota 23 could be directly affected based on these large shifts in FASAs that are seen. This is also that time where we see that extremely high outlier. And so not only is it a shift in abundance, but it's a really large shift in the total flux that's being observed at this site. And so Minnesota 16, on the other hand, we're really not seeing those FASAs appear, but we are seeing the sulfonic acid PFAS starting to increase. And so this could be either atmospheric transformation or even biotransformation of these PFAS through the growing season, and then they get kicked up during harvest or reapplication of biosolids. This is still speculation at this point, and so I need to do a little more work to verify if this is the only source or the most likely that we're seeing. So bringing all the data back together here, the FASAs aren't the only noteworthy changes throughout the year. We do see this increase in Gen X in Michigan in the summer. And so I'm starting to think about, is this either manufacturing practice shifts throughout the year? Is it just those increased temperatures making these PFAS more volatile? I'm not quite sure yet. I'd love to hear ideas, but this is where I'm moving forward with this data. And we are continuing to sample through July of 2025, and I'll be using positive matrix factorization, which is a source receptor model that is used to elucidate source types as well as source contributions for each sample. And like I did with the biosolids, I am using current literature to find these likely sources of our observed analytes. All right, so let's continue on and start talking about lake sediments. And I am specifically not looking at the Great Lakes because they're really large and they have very complex inputs and mixing mechanisms that it's difficult to start off with. So we collected a lake sediment core in northern Minnesota last summer. And the purpose of this lake sediment was to get an idea of the baseline atmospheric inputs of PFAS into lakes. And so Lorain Lake is a precipitation-dominated seepage lake. There are no rivers or streams discharging into it, and it just has very low development within its small watershed. So what we're seeing in this lake sediment core is fairly small amounts of PFAS, but they're detectable and they're there. And it is similar to what's seen in other secluded lakes, where we just have this general increasing trend of more PFAS in the more recent sediments. And so of those PFAS, we're seeing again, a majority are carboxylic acids, which is similar to our wet deposition observations, but they're not exactly the same. Comparing those most recent sediments, this red line is where I cut off the sulfonic acids and the carboxylic acid PFAS, and we can see that it's about 75% of what we're seeing in the sediments, where in the wet deposition, it's almost 90. And so of those PFCAs, we're seeing a large difference between the long chain in the sediments versus the short chain being more abundant in the wet deposition, which I have outlined here in the dashed red box. Those are our long chain species. And so this feels like a lot of differences, but I'm going to add another one into it. The estimated flux that we're seeing at these sites is almost an order of magnitude different when compared to the wet deposition. And I say almost an order of magnitude different because our more closely situated precip site is, quite a bit lower. So I specifically added Minnesota 23 in here because Lorain Lake is in a more densely populated area, so it's likely to receive a little bit more PFAS. And on top of that, we did not see much precipitation in Minnesota in the spring, so it could be that this year was just lower on average. So where are these PFAS going and why are we seeing such different profiles in the wet deposition versus the sediments? Some possible explanations for a reduced total PFAS flux could be general partitioning. Some PFAS could be re-emitted back into the atmosphere, while others are retained in the water column or even uptaken from biota within the lake. And this could also impact the amounts of, or types of PFAS that we're seeing. Long chain ones and sulfonates generally are more sticky and like sediments, but we could also be seeing some preferential partitioning due to winter. And so Lorain Lake is in northern Minnesota, it freezes over, and we see a lot of snow up here. And so there have been some more recent articles that show that there are effective enhancing or retention of these long chain PFCAs and sulfonic acid PFAS during the formation of ice, and as well as when snow is sitting out and the short chain PFCAs are able to escape or be effectively excluded during this time. So whether or not this is a large impact on the profile of the lake, the profile of PFAS within the lake, is still what I'm looking at. Is there one of these fate and transport mechanisms that is controlling these differences? So that is where I'm moving on with that. And with the last couple minutes that I have here, I'm going to bring in some of these Great Lakes, specifically Lake Superior, because that's the data I have, and it's our more precipitation-dominated lake. And so this data was collected in part of the Great Lakes Sediment Surveillance Program and was analyzed here in Duluth at the Natural Resources Research Institute. And so all of these different sites on the lake are our lake sediment grabs. And the bar graph on the right-hand side is all of the different sediment sites, their PFAS distribution. And so you can see that it's kind of split between the profiles. We have these ones that I've called the offshore sites that have a pretty big distribution of different PFCAs, while the nearshore sites are more sulfonate dominated. And so this is also related to the total amounts of PFAS within the lake. These offshore sites generally have higher amounts of total PFAS where the nearshore sites are lower. And so this lends to the idea that Lake Superior has a more atmospherically dominated profile because we see 75% of these PFAS are PFCAs. But let's compare this with our atmospherically dominated lake a little more directly. So here I've pulled together just the top interval of Lorain Lake along with 2 nearshore and 2 offshore sites, as well as the highest contributing PFAS within the sediments. So just looking at the data briefly, Lorain Lake, our light bar versus our 2 nearshore, the purple and black, kind of mimic each other between the analytes, excluding PFBA and PFBS because they're very mobile and weird. But so our atmospheric lake looks like a nearshore Lake Superior site is kind of what I've gleaned from this. This is pretty new to me, but some easy explanations that I could think of is that Lorain Lake has a relatively high sediment mass accumulation rate, which is similar to what is in the nearshore sites of Lake Superior, which is, you can kind of relate to the percent sand. And so this could be effective sediment dilution or some other process that I'm not familiar with yet. Or this could be, again, ice cover, where we're seeing the center of the lake rarely freezes over, and these nearshore sites could be more impacted by that preferential partitioning throughout the year. So moving along with these 2 data sets, which I said is pretty new, so any insights or advice anyone has, I would greatly appreciate. I'm going to continue to compare those lake dynamics and the potential sources impacting the 2 lakes. So wrapping up with conclusions, wet deposited PFAS, like we've heard all morning, lots of PFCAs, but there are these significant events that could indicate site-specific sources. And then atmospheric lake sediments, again, primarily PFCAs, but there are these differences in the profile and estimated fluxes that could be due to partitioning or preferential partitioning. So with that, I'd like to thank everyone for listening, and I'd be happy to take any questions.
Questions?
Thanks for your talk. You had one slide that showed like wind direction as a general vector. Have you looked at any like back trajectory, forward trajectory models of where the air masses are like distributed coming from and where they're going? Like what percentage of them actually go over the lakes?
Not yet. That is a part of this project that I unfortunately won't be here for, but I think that is happening maybe next year-ish or the year after, so stay tuned. Because they will be looking at these more like unique events to see where the sources could be. Yeah.
Great, thanks.
You have a question, Alex? Oh,
Yeah, so I have a quick question. So we've heard from Marta and from Alicia, Allie. Sorry, we've heard from Marta and Allie about how their sites were showing very similar concentrations between sites, but your graph showed differences. Can you talk about a little bit how you presented the data slightly different? Concentrations versus fluxes?
Sure. So I tend to use fluxes because concentrations for PFAS samples can be a little weird with rain because, I don't know, the concentration and the amount of rain within the sample. For ours, we are seeing like as the more rain that happens, the less the concentration is, which is essentially the inverse of that flux graph that we saw. So we're seeing dilution of the PFAS with higher rain events. So that's why I like to stick with fluxes instead of concentrations, but the 2 are kind of similar.
I do have one comment online if we have time. It's from Twin Ports Freshwater, and it just says biosolids solutions ?
Well, I think so. Biosolids and wastewater treatment plants and PFAS altogether is a really complex issue because it can vary from facility to facility and, state to state. But the Minnesota Pollution Control Agency has this this PFAS blueprint that is going on right now, which incorporates looking at source identification and reduction work within wastewater treatment plants. So I think they are in the right direction because you can't just get rid of the PFAS that are in the biosolids because it's such a nasty matrix. And destruction of the PFAS in biosolids is not really an option, like cost-wise, I think. So yeah, source identification and reduction is usually your best bet for PFAS stuff.
Hello, everyone. Thank you for tuning in to my pre-recorded presentation. I'm Daniel Prasad, a current graduate student at York University, and today I'm excited to share some of the work we've been doing to understand the occurrence of peripheral alkyl substances in Newfoundland and Labrador, Canada. Before we begin, just a brief introduction. PFAS describes a class of millions of anthropogenic compounds with at least one CF2 group. This is actually a very broad definition that describes several different families of compounds that have different chemical properties. They could be further classified as being perfluorinated, where all carbons are substituted with fluorine. These are usually very persistent and unreactive. Here we have an example of the very popular perfluorooctanoic acid, or C8. The polyfluorinated compounds, on the other hand, may have other substituents, such as hydrogens. These are usually more reactive. This class of compounds are very important to us since polyfluorinated compounds can be precursors to the more persistent perfluorinated compounds, something that I will get into during the presentation. These PFAS compounds are unique since they can have both hydrophobic and oleophobic properties and are far superior when compared to their hydrocarbon counterparts. This led to extensive use as performance chemicals in non-stick and fire suppressant technologies. The perfluorinated compounds can be further classified by their functional groups. The two most popular groups are the perfluorocarboxylic acids and the perfluorosulfonic acids, which are further classified based on their chain length. The PFCAs and PFSAs are collectively referred to as perfluoroalkyl acids, or PFAAs. So although these compounds provide useful applications, some concerns have been raised over the years. For one, they are extremely persistent in the environment. We can find these compounds in every sample that we look in. They can bioaccumulate in aquatic and terrestrial food webs. They are toxic. It is well known that long-chain PFAS has endocrine disruptive properties and are possible carcinogens. And they can undergo long-range transport, which is not expected by this class of compounds. These are extremely strong acids, and most of the deposition is expected to be very localized, but we know that that's not always the case. So for this campaign, we were trying to answer a few science questions. Firstly, we were interested in discerning which PFAA homologs are present in the Newfoundland and Labrador region. Also, how does the deposition vary in space and time? Is the dry deposition mechanism an important removal pathway? And what are the main sources of PFAAs in this region? Is it localized direct transport, or is it indirect formation in the atmosphere? To answer these questions, samplers were deployed across 4 sites in Newfoundland and Labrador. They are Grand Cordroy, Humber River, Salmon River, and Eagle River. These sites are located on the only latitudinal transect in Canada, where temperature and precipitation decrease as latitude increases. We collected monthly precipitation samples from October 2013 to August 2016. We had 2 different types of samplers at each site. To collect total deposition, which is wet plus dry, we utilized a PVC pipe with an internal diameter of 0.23 meters. We also deployed our custom-built off-grid automated samplers to collect wet-only deposition. Here is a little video of how our wet deposition sampler work. These samplers will automatically open when a rain sensor detects conductive precipitation and closes after a precipitation or washout event. If you would like more information on the operation and validation of these samplers, please scan the QR code that links our paper. Now moving on to some results. We detected at least one PFCA from C4 to C14 in precipitation samples. We see the usual high detection of the C8, C9, and C10 homolog, followed by a subsequent decrease. PFOS was the most detected PFSA, followed by a decrease from PFBS to PFHBS. In terms of observed concentration, the median PFCA concentration was 300 picogram per liter, with values ranging from below LOD to 10,000 picograms per liter. We can see that PFBA accounted for most of this. The PFSAs were detected with concentration an order of magnitude lower than the corresponding PFCAs, with a median concentration of 3 picogram per liter. We found that PFOS and PFBS were detected in almost equal quantities, with concentration ranging from below LOD to 200 picogram per liters. Now, concentrations are not very useful in determining trends, so we converted measured concentration to deposition fluxes using volume, area, and number of sampling days. We found that the average PFCA flux was 500 nanogram per meter squared per annum, whereas the average PFSA flux was 13 nanogram per meter squared per annum. But we wanted to determine if any latitudinal differences exist between the sites. So we utilized the Tukey's HSD and the Newman-Kulis test to determine if there's any statistically significant differences between the sampling locations. We found that the null hypothesis was accepted for the C5 and C8 homolog and the C10 to C14 homologs. This suggests that no significant differences exist between the sites. The C4 and C9 homologs were exceptions. We found that differences exist between the northernmost site and the three island sites. This is somewhat expected since we expect the fluxes to decrease as we increase latitude. To determine the dry deposition contribution, automated samplers were deployed simultaneously with total deposition samplers at each site. The dry deposition contribution was then estimated by subtracting the PFAA fluxes in wet deposition from total deposition. What we found is that the dry deposition mechanism was a predominant pathway for the removal of PFAAs in this region. The striped bar represents the estimated dry deposition contribution. We can see that fluxes range from 2100 to 3200 nanogram per meter squared per annum for PFBA and between 100 to 400 nanogram per meter squared per annum for PFOA and PFNA. This suggests that the dry deposition is an important removal mechanism of PFAAs. Now I would highlight some transport mechanisms that may be responsible for the observed deposition in this region, starting with answering the question, is direct transport by marine aerosol a major contributor? So correlation with major ions is one tool we use to determine if direct transport is a major contributor. We correlated flux with measured sodium, non-C-spray calcium, magnesium, and potassium as tracers of C-spray, mineral dust, and biomass burning. The strength of the correlation is highlighted by the color and the size of the circles, and a black outline represents a statistically significant correlation. We found that most PFAAs were not statistically correlated with major ions. The lack of sodium correlation was also surprising for us since these are all coastal sites, and if C-spray was a dominant pathway, then we would expect most PFAAs to be correlated with sodium, but we only saw two statistically significant correlations. At Grand Carteret, the C10 homolog was correlated with sodium, and PFBA was correlated with sodium at Humber River. To further determine if local sources are important, we generated back trajectories for each integrated sampling period using NOAA's high-split model. The trajectory endpoints were then compiled and assigned to one of four sectors, either northeast, northwest, southeast, and southwest. We then compared the deposition to the sectors to determine if any relationship exists. Here's an example at Humber River. We can see that there is no real clear trend And this highlights the complexity of using back trajectories to try to explain the observed deposition. Another mechanism that could lead to the deposition of PFCAs is the atmospheric oxidation and transport of volatile precursors. Now, it is well established that fluorothermal compounds are major precursors of PFCAs. They can be produced with even number of carbons, such as the 8,2 and 10,2F2s. The OH-initiated oxidation leads to the formation of the perfluorinated aldehydes. There are two main mechanisms that form PFCAs from the perfluorinated aldehyde under low NOx condition. In the first pathway, O2 is added to the terminal carbon, whereas in the second pathway, CO is removed from the perfluorinated aldehyde. We use these homolog patterns as fingerprint of atmospheric oxidation. If these sources are important, then we expect equal amounts of pairs of PFCAs. We calculated the oxidation products of the 10,2, 8,2, 6,2, and 4,2F2s, which should produce PFCAs from C4 to C11. We found that the longer chain oxidation products are within a factor of 2, with most within a factor of 6. The opposite trend was observed for the PFBA-PFPEA pair, where almost all of the molar ratios are not within a factor of 6. This suggests that the oxidation of volatile precursors are not a major source of PFBA in this region. We also correlated individual PFAs to determine if any relationship exists. The idea is if these compounds are coming from the same source, then sequential pair concentrations are expected to vary through time together. And that's exactly what we found. Most PFAs produce a statistically significant correlation with each other, which further confirms our hypothesis that most PFCAs are formed through precursor oxidation. Finally, we compared our observed deposition to our recent geoschem model. This is an updated chemical model that includes PFCA formation from thermal oxidation. We found that the model could account for more than 70% of the C5 to C12 PFCAs. The greatest disparity was observed for the PFBA homolog, where the model could only account for 5 to 10% of the observed deposition. All of the evidence thus far suggests that PFBA is not formed from precursor oxidation. Other sources of PFBA could be the oxidation of CFC replacement compounds. Now, the key takeaway from this presentation, PFCAs and PFSAs were detected in at least one sample. Most PFAs did not show any latitudinal differences. PFBA and PFNA were notable exceptions. The dry deposition is an important removal mechanism for selected PFAs. There is limited evidence for direct transport by sea spray, mineral dust, and biomass burning, and most PFCAs are formed via oxidation of volatile precursor. That brings me to the end of my presentation, but before I go, I'd like to thank everyone that supported this project, and thank you for your attention, and I'll take any questions. Thank you.